Hello everyone, welcome to my channel GVS Chem. In this video, I am going to explain enzymes and vitamins, the deficiency of vitamins and the sources of vitamins. Also, some multiple choice questions in the chapter Biomolecules. Please like, share and subscribe to my channel. Enzymes are the complex nitrogenous organic substances which are nothing but proteins which are produced by living plants and animals. During the digestion of the food, the different molecules are involved in various reactions. The process of digestion involves a sequence of reactions occur in the body under very mild conditions. These reactions occur in the presence of catalysts. Do you know catalysts? What are catalysts? Catalysts are the substances which alter the speed of the reaction. Alter means the speed may increase, the speed may decrease. Since these reactions occur in the living body, these catalysts are referred as biocatalysts, and of course these biocatalysts are referred as enzymes. Now, let us have some examples of enzymes. So, we have sucrose, C12H22O11. The sucrose in the presence of enzyme known as invertase. So, in the presence of invertase, it decomposes into glucose C6H12O6 plus fructose C6H12O6. So this is sucrose. What is sucrose? Cane sugar. This is glucose. This is fructose. Both glucose and fructose have the same molecular formula. Therefore, they are isomers. These are optical isomers. Now, this glucose and fructose C6H12O6 in the presence of enzyme known as zymase. In the presence of zymase, we get C2H5OH plus CO2. So this is a conversion of cane sugar into ethyl alcohol. So this is the reaction involved in the manufacture of alcohol in distilleries. This sucrose is nothing but the brown colored sucrose. Here the sucrose is nothing but the brown colored molasses which is left after the extraction of sugar in sugarcane industries. So this sucrose converts into glucose and fructose in the presence of invertase, and this glucose and fructose converts into ethyl alcohol in the presence of enzyme known as zymase. Right. Now another enzyme is C12H22O11, and here the enzyme is maltase plus H2O. We get in the presence of maltase. So this sugar is maltose here. So we get C6H12O6. This is glucose, and we get two molecules of glucose here. So maltose in the presence of enzyme maltase, we get glucose. And another reaction is C6H10O5N. What is this substance? This substance is starch. NH2O in the presence of a catalyst diastase. So in the presence of catalyst diastase, we get maltose. C12H22O11. This is maltose. Of course, these reactions take place in the human body. So here, starch converts into maltose, and of course, then maltose converts into glucose. And of course, this glucose dissolves in the blood, and during the respiration, this glucose combines with oxygen to form carbon dioxide, and energy releases, and this energy we use for different day-to-day activities. NH2CO, NH2. What is this substance? This is urea. Urea, in the presence of catalyst urease, urea in the presence of catalyst urease and converts into ammonia, NH3, and of course CO2. So this here the enzyme used is urease. Enzyme pepsin converts proteins into peptides. Trypsin, another enzyme, trypsin, produced in pancreas. Trypsin produced in pancreas converts proteins into nucleic acid. Proteins converts into amino acids. And another enzyme, lactobacilli. An enzyme converts milk into curd. So these are the different enzymes that are involved in different reactions, that is specific reaction. Each enzyme is specific in its action. That is the enzyme urease that converts urea into ammonia and carbon dioxide. Enzymes are mostly globular proteins. We discussed already in the previous video, that is proteins are of two types. They are specific for a particular reaction and a particular substance. Mechanism of enzymatic catalysis. There are the number of cavities. Number of cavities means the holes or the specific grooves on the surface of the colloidal particles of the enzyme. Colloidal particles means the particles of the colloidal substance varies in the size from 100 nanometer to 1000 and more nanometer size. These cavities are the characteristic shape and possess active groups like carboxy group, amine, thioalcohol, alcohol, etc. These are actually the active centers of the particles of the enzyme particles. Active centers means at these centers the reaction takes place. The molecules of the reactant, also referred as substrate, which have a complementary shape, fit into these cavities. Complementary shape means whatever be the shape of the cavity present on the catalyst, in that cavity, the substrate molecule or reactant molecule exactly fits. That is just like a key fits into the lock. So here this is the enzyme, and this is the substrate. The substrate has a groove in the particular shape. In the same shape the catalyst has. So this catalyst both have the complementary shapes. Therefore this enzyme can exactly fit into this substrate. Therefore, and of course this is referred as active site. Active site means where the reaction of the substrate or reactant takes place. So this enzyme fitted here, and of course once the enzyme fixes into the substrate, the reaction takes place and product is formed. So these are the product molecules or product particles, and of course enzyme came back. So thus the enzyme catalyzed reactions are considered to proceed in two steps. In step 1, catalyst fits into the active site of the substrate to form activated complex. In step 2, the decomposition of activated complex to form product, that is again catalyst, enzyme, that is catalyst comes back. Now vitamins. Vitamins are the organic compounds required in the diet in small quantities to perform specific biological functions. for normal maintenance of optimum growth of the health of the optimum growth and health of the organism. Classification of vitamins. Vitamins are classified into two groups depending on their solubility in water or fat. The first one is fat soluble vitamins. Vitamins which are soluble in fat and oils but insoluble in water are kept in this group. These vitamins are A, D, E, and K. They are stored in the liver and adipose tissues. The second category is water soluble vitamins. B group vitamins and vitamin C are soluble in water, so they are grouped together. Water soluble vitamins must be supplied regularly in diet because they are readily excreted in urine and cannot be stored. So this is a common experience of individual, that is whenever beetroot is consumed, so after few hours you can observe the color of the beetroot in urine coming out. That is, the vitamin present in beetroot is water soluble, therefore it comes out. So water soluble vitamins go out regularly through urine, therefore they should be supplied from time to time. Now let us see the types of vitamins. Vitamin A, it is found in fish liver oil, carrots, butter, milk. And the deficiency disease, as vitamin A is absent, is not present in our diet, we get a disease referred as xerophthalmia, that is hardening of cornea of eye. Due to this, the vision becomes less. Vitamin B1 present in yeast, milk, green vegetables and cereals. And the deficiency of this vitamin causes beriberi means loss of appetite, retarded growth, etc. And cheiliosis, fissuring at corners of the mouth and lips, digestive disorders and burning sensation. Vitamin B2 present in milk, egg white, liver and kidney. Vitamin B6 present in yeast, milk, egg yolk, cereals and grams, different grams like green gram, Bengal gram, etc. And the deficiency of this vitamin B6 causes convulsions, that is fits. Vitamin B12 present in milk, egg, fish, curd. And the deficiency causes pernicious anemia, that is less amount of red blood cells in the blood. Vitamin C is also referred as ascorbic acid. This is present in citrus fruits like lemon, etc. Amla, that is tamarind, green leafy vegetables. The deficiency of this vitamin causes scurvy, that is bleeding of the gums. Vitamin G, exposure to sunlight, fish, egg yolk. The absence of vitamin D causes rickets, bone deformities in children. Bone deformity means bones are not present in proper shape. Vitamin E, vegetable oils, present in vegetable oils like wheat, gram oil, sunflower oil, etc. Increased the deficiency of vitamin E causes increased fragility of RBCs, means red blood cells, and muscular weakness. Vitamin K, sources present in green leafy vegetables, and deficiency causes increased blood clotting time. Now let us discuss some questions. Optically active polyhydroxyaldehydes or ketones are their compounds which give units of C6H12O6 are called. The correct choice here is carbohydrates. Correct choice here is carbohydrates. So option B. Fructose is a ketohexose. Fructose has a functional group ketone, and of course it has 6 carbon atoms. Therefore it is a ketohexose. So the correct option is A. Third question. Sucrose on boiling with HCl to give 2 molecules of glucose, 2 molecules of fructose, 1 molecule of glucose and fructose, 1 molecule of glucose and galactose. The correct option is sucrose is a monosaccharide with glucose and fructose. Therefore, the correct option is C. Fourth question. Glucose is one of the monomers of sucrose, maltose and lactose. All these. Sucrose is the disaccharide of glucose and fructose. Maltose is the disaccharide of 2 glucose units. Lactose is the disaccharide of glucose and galactose. Therefore, the correct option here is all these. Option D. Glucose on prolonged heating with hydrogen iodide gives N-hexane. The correct option here is out of N-hexane, isohexane, neohexane and multi-branched hexane. So the correct option is N-hexane, that is normal hexane. That is all the 6 carbon atoms are present on the single chain. So the option here is A. On oxidation, question number 6. On oxidation with nitric acid, glucose gives adipic acid, saccharic acid, glutaric acid and malonic acid. The correct option here is B, that is saccharic acid. So the correct option is saccharic acid. 2 configurations of glucose forms which differ in C1 carbon are called epimers, anomers, both of these, none of these. The correct option is anomers. Option B. Question number 8. Glycosidic linkage between glucose and fructose in sucrose is in between C1C1, C2C1, C2C2, C1C2. The correct option here is option D, that is in between C1C2, that is C1 of glucose and C2 of fructose, they join to form glycosidic linkage. Therefore, the correct option is D. Milk sugar is the sugar present in milk, sucrose, maltose, lactose, all these. The correct option is lactose. Lactose is the sugar present in milk. So the correct option here is C. Which is non-reducing sugar? Which is non-reducing sugar? Sucrose, maltose, lactose and all these. Non-reducing sugar is that which doesn't reduce the tolerance reagent and Fehling's solution. In sucrose, C1 of glucose and C2 of fructose both are reducing carbons and they are involved in glycosidic linkage. Therefore, sucrose is a non-reducing sugar. Therefore, the correct option is A. The hydrolysis products of lactose are 2 units of alpha D glucose, 2 units of beta D glucose, 1 unit of beta D galactose and 1 unit of beta D glucose. So the correct option is C, that is 1 unit of beta D galactose and 1 unit of beta D glucose. So the option here is C. Now question number 12. Which of the following is essential amino acid? Alanine, glycine, valine, glutamic acid. Valine is essential amino acid, that is which should be supplied through diet. Therefore, the correct option is C. So, you can practice all these questions and prepare for the examination.